Fantastic. Well, our final session of the afternoon is actually one of the more exciting ones because everybody likes to ask about genetics and BRAF and personalized medicine. Dr. Fagin is the chief of the endocrine service here and is one of the world's real experts on understanding the molecular biology of thyroid cancer. He's got a tremendous lab. A lot of the stuff that Dr. Sherman was telling us about this morning about the various drugs and the trials is a very close interaction between Dr. Fagin's lab and the medical oncology group, so really putting some focus on how we do things. In addition, Dr. Fagin's also a clinical endocrinologist, so he still sees patients with us as well. So he has this unique ability to have a foot in both worlds, both being a really excellent clinician and also really a world-class lab person. So we are thrilled that he's come spend with us and talk to us a little bit about whatever the heck the word genomic landscape means. Jim?
I'm always afraid of any introduction from Mike Tuttle because you never know what's going to come here. But it could have been worse, let's put it that way. So anyway, it's a pleasure to be here. And what I thought I was going to do is to hopefully give you enough background so that this entire revolution that we're seeing in our understanding of the molecular base of thyroid cancer and of cancer in general becomes understandable to you, and also that you see the implications, how we're trying to project this and make it useful for patients, both in terms of improving how we diagnose the disease as well as how we treat it. So the first thing to state, and I think something that everybody is aware of already, is that cancer is really a genetic disease. It's caused by genetic changes. We call these mutations. And by and large, they tend to be mutations that we acquire during our lifetime. So if a cell normally has its complement of genetic material, over the life cycle of that cell, as it divides, it has progeny, as we age in particular, errors happen when the DNA is replicating, okay? And those errors are called mutations. They happen when the cell divides. Most of those mutations are innocuous. They just sort of get carried along and ultimately just fizzle out because they don't give the cell any particular advantage. But if the mutation in the gene actually changes the biology of the cell in such a way that it makes that cell grow disproportionately, then that's the first step towards developing a neoplasm. And in most cases, I mentioned, this happens after we're born. There are certain situations where cancers can be inherited because there's genetic changes that are in our germline that favor or predispose us to have certain types of cancers. One that you mostly will be familiar with is, of course, the inheritance of breast cancer that happens through certain susceptibility genes. In thyroid cancer, we know that it can run in families. Maybe about 5% of papillary thyroid cancers actually happen clustered in families. But there doesn't appear to be a single gene that we can put our finger on and say, okay, this particular gene is conferring that predisposition for that family to have a greater frequency of thyroid cancer. So by and large, we think of this as a sporadic disease. It happens randomly, these genetic changes, in a way we don't know what caused them. Sometimes radiation can be a factor that causes a mutation, and the example of the Chernobyl nuclear reactor accident, where the children who were exposed to radiation then had a much higher incidence of thyroid cancer, that was a clear example of how physical damage to our DNA can actually result in predisposing to a particular disease, and thyroid cancer specifically. We understand the basis for that, but that's rare. In most cases, people get thyroid cancer and we cannot explain why they got it. However, we do want to identify the genes that cause the cancer. First, we need to understand it in general terms. So if we take many, many different thyroid cancer specimens, are there particular genetic changes that happen more frequently than others? And that's been something that many groups have been involved with for many years to try and understand, including our own. And we'll talk a little bit about how we used to do it and how we actually do it now. And it's a revolution in actually our ability to pick up and detect these abnormal genes. So one way of thinking about it is maybe it can help to look at this slide. So what you're looking at here is this hypothetical cell that I sort of made up. What these structures here are supposed to be chromosomes, and I put a little asterisk here, and that asterisk represents a mutation in a particular gene, and that happens to give this cell a growth advantage. And as you can see here, it's perpetuated. These two daughter cells carry the same mutation. If at some point one of these cells acquires a second mutation, and that one gives it an even greater growth advantage, then it will begin taking over the entire cell population, dominate that cell population, and expand. And eventually, if a third mutation happens, as illustrates here, then this new cell will be the dominant clone. And this is what we call tumor microevolution. It starts with an initiating event, then there can be other genetic changes that accumulate, and over time, as that genetic burden increases, the cancers become more worrisome, okay, because they have more genetic changes. So why is it then important to identify the specific genes that are mutated in any given cancer? Well, one important concept is this concept of cancer cell addiction. And what this means is that the cancer cells become dependent, that is addicted, to the oncogenic driver. So let's imagine that a mutation, what it does is it changes the activity of a growth factor receptor, and it makes it tell the cell, you've got to divide. And that happens constantly and never stops. The cell ultimately becomes totally dependent on the activity of that abnormal gene. It usurps the growth control mechanisms of that particular cell. That creates a dependency that we can exploit. And that's the overall basis of this, what we call this Achilles heel of cancer, that if you understand where the mutations are, and you can then understand how they signal and change the biology of the cell, you then have a therapeutic target. You have a place where if you get a drug that can block the activity of that mutated protein, then that actually can result in very important and beneficial effects for patients. And there was a very important example, like a proof of principle of this, that happened 10 or 15 years ago, which was the discovery of drugs that could block the activity of a particular leukemia gene that causes chronic myelogenous leukemia. And this drug, which is called imatinib or Gleevec, blocks the activity of this genetic mutation in a gene called ABL, and that changed the life of patients with chronic myelogenous leukemia that nowadays is a very treatable disease associated with much longer survival. And that became sort of a mantra that many of us wanted to follow and say, well, can we do this for the disease that we want to treat? And that's what we've been trying to do for thyroid cancer as well. Besides helping us identify targeted therapies or where we should be thinking about targeting a disease, if we understand the genetic base of the disease, it also gives us genes that we can sequence, and these could actually aid in the diagnosis of the disease. So we'll talk a little bit about the fact that normally when somebody has a nodule and that nodule is aspirated, a biopsy is done, and we look at the cells under the microscope, we often can tell whether the tumor is benign or whether it's malignant. But sometimes there are these equivocal gray zones where we're not sure. And these genetic tests can actually clarify that. And I'll talk a little bit about that in a second. So what is the genomic landscape? Well, it seems like a very big word, like Mike was saying. So sort of before, when I started in this field 25 years ago, and we wanted to identify genes involved in thyroid cancer, it was like a needle in a haystack. We essentially had to think of candidates. So if somebody found a gene that was abnormal in a different type of cancer, I would just say, well, maybe this happens in thyroid as well. That's what we would call this candidate gene approaches. So you would, in a sort of arbitrary way, try and identify genes by sequencing very short stretches of DNA. We made some progress with that. It took a long, long time. But now, since the sequencing of the human genome, now we can actually sequence cancer genomes very inexpensively. So whereas before it took billions of dollars to sequence the germline DNA of one individual, now we can actually, for $1,000, sequence cancer genomes and try and understand the genetic makeup of a particular cancer. So there's an entity in the National Cancer Institute that is sponsoring what's called the Cancer Genome Atlas, called TCGA. And TCGA is trying to do definitive sequencing studies for all major cancer types. And 2 months ago, or 3 months ago, there was a paper published in Cell with the genomic landscape of thyroid cancer. And I'm going to tell you a little bit about that. So as I mentioned, the potential clinical impact of doing this is that it can help us differentially diagnose thyroid nodules. It can improve our ability to risk stratify. And you've heard a little bit about that in the clinical talks this morning. But right now, we are still categorizing risk based on the size of the tumor, the presence of lymph nodes, the appearance of the cells under the microscope. But what we're learning is that the genetic base of the disease tells us a lot about how that tumor is likely to behave in the long term. So it could improve us to risk stratify. We'll talk a little bit about how it can also help us to understand which patients benefit from being on higher doses of thyroid hormone in order to suppress TSH, which commonly is done in this disease virtually for everybody. We have an inkling that maybe we don't need to do it for everybody because some people might not benefit from that. And the genetic studies are telling us this. And finally, as I mentioned before, we try and identify these Achilles heels of different cancers, which may differ between different individuals with thyroid cancer so that we can personalize therapies and give them a drug that will specifically interact and block the activity of this oncogenic event. So this is the Cancer Genome Atlas. Just to give you a sense of the magnitude of the effort here, these are all groups at different universities and institutions around the country. Each one of this is doing a different piece of this project, which are 500 papillary thyroid cancers, the most common form of thyroid cancer, that were sequenced. The DNA was sequenced. The messenger RNA, which is essentially what is transcribed from the genome and sent as a message to make protein, that was sequenced too. The protein characteristics, so very complicated set of genomic analysis. And we call it a genomic landscape because you're really looking at the entire picture. It's not just one or two candidates. You're looking at the whole thing. So, as I said, it was 500 clinically and pathologically annotated papillary thyroid cancers, and Memorial Sloan Kettering was a very important contributor to this effort because many of us participated in this, and also we were fortunate that we could also donate a number of samples that were analyzed by this group. So what have we learned? So what you're looking at here is there's now been efforts by the Cancer Genome Atlas to sequence many different cancer types. And here you see the different types of cancers that have been done. You can see acute myeloid leukemia, chronic lymphocytic leukemia, kidney carcinomas, and so on, virtually every type of cancer. And what you're looking at these black dots are how many mutations were found in the genomes of these different cancers. And there are some, and this is a logarithmic scale, meaning that there's actually the magnitude of this is more than 10 times what you're seeing here. Some cancers have a very, very high mutation burden. And if you remember for that early slide where I showed you these little pictures of cells with chromosomes and mutations that accumulate over time, this means that these particular cancers are loaded with genetic changes. And these are the bad actors. This is melanoma, for instance, that in the advanced stages of very lethal disease. These are full of mutations because ultraviolet light from the sun damages the DNA. Thyroid cancers, fortunately, have a very low frequency of mutations. And that's good, and also explains why, by and large, this is a disease that is not so severe and is indolent and manageable because it usually has very few genetic changes. And this really complicated slide, I'll try and explain it for you. Each one of these little bars here, or columns here, represents the DNA of one patient. And where you see these little blue areas here means that a mutation was found in a gene, and right here is the actual name of the gene that you probably can't see very well, but this says BRAF, this says RAS, etc. And as you can see, the most common mutation in thyroid cancer is BRAF, something that actually our lab discovered many years ago. Many others also found the same thing. It's about 60% roughly of all thyroid cancers have BRAF mutations. In addition to that, there are mutations of another gene which is part of a signaling cascade which is called RAS. And finally, there are mutations in these receptor tyrosine kinase fusions. And so what you can tell is that these mutations don't overlap, right? If a tumor has a mutation in BRAF, it doesn't have any of the other driver mutations. Why? Because this mutation actually is the one in charge to which the cell has become dependent. Okay? And so this, in a way, is telling us what the drivers, who is in control of each different cancer type. And by doing that, it tells us what we need to target and how we have to think of this particular cancer. So in what way is this already changing things? Some of you may have heard that when we have biopsies of nodules and we're not able, just by looking at the cells under the microscope, to distinguish whether they're benign or malignant, there's a gene expression classifier which basically looks at the actual messenger RNAs that a nodule is expressing, a cancer cell is expressing, to try and determine whether it's a benign or malignant tumor. And this test is designed to have a very high efficiency at telling you whether that nodule is benign. We call that a high negative predictive value. Okay? So if your gene expression classifier, it's called a FIRMA, tells you this is benign, 95% of the times it really is benign. And we can reassure our patients, maybe we can watch this. This is a tumor that probably we don't need to go to surgery with. Okay? But it doesn't have, it's not very good at telling you which is actually a cancer. Based on the findings of the TCGA, what we can now do is have a test that has a high positive predictive value. And the reason is that if you go back here, if we add up these mutations of BRAF and these mutations of RAS and some other rarer ones, plus all these receptor tyrosine kinase fusions and a few rare ones that are over here as well, we actually come close to 95%. And now you can actually make panels to detect in the DNA of the fine needle aspiration biopsy which of these mutations are present. And if they're there, it tells you this is likely to be a cancer. And it tells you, well, this patient needs to go to surgery. So just by this TCGA effort, it's allowed this particular test, which is called ThyroSeq, and it's done by an investigator at the University of Pittsburgh, Dr. Nikiforov, put this panel together. These in yellow are the genes that he added to his panel after the Cancer Genome Atlas published this paper, okay, because it completed the picture of the genomic abnormalities in this disease. We suspect that this particular test now will become a dominant way in which nodules are diagnosed when we're not certain after just looking at it under the microscope. Okay, so I apologize for showing you lots of different original data slides from a paper, but I'm hoping I can make this understandable for you. Here on top again, what we're looking at is the genes that are detected in these different tumors. Again, here you have the BRAF mutation. These mutations here are the RAS mutations and the receptor tyrosine kinase fusions. What I'm showing you this for, the purpose of this, is to try and see how this connects with the ability of a tumor to trap radioactive iodine. Okay? And if you just look here, you can see all of these patients had BRAF mutant thyroid cancers. And what we're looking at in this panel below is which of the messenger RNAs for the different proteins that are required to trap and incorporate iodine into tumors. Everything that is in green means that the expression is low. Everything that's in red is high. And you can immediately see that the BRAF mutant tumors predominantly express very low amounts of the genes that are required to trap iodine. Okay? By contrast, those that have RAS and receptor tyrosine kinase fusions are actually quite a lot better. Okay? There's an exception. There's a little sort of cluster of BRAF tumors that retain these differentiated properties. We're very interested in understanding why. So where are we going with this? Well, for instance, what we're now doing at our institution is, as a research project, is we're developing a particular panel that will be able to look at this green-red difference before patients are actually targeted to give radioactive iodine or not, so we can see, is this really predictive? Does this tell us which patients are likely to benefit, and in which patients they'll just get the toxicity, but they won't get the benefit? Okay? So now we have a gene that we already know, BRAF, that mostly, but not always, is associated with tumors that don't trap iodine well. There's a subset of them that do, and we want to be able to understand who are these patients and why are they different. And then there are others that actually should do quite well with radioactive iodine, which are these with the different mutations I mentioned before. So from this big genomic landscape study, we have a number of leads to begin investigating and refine, personalize, who could be given radioactive iodine and who doesn't need it. So, just recapitulating, based on what we now know from this Cancer Genome Atlas study of papillary thyroid cancer, the other thing that we can do is to try and see who can benefit from high doses of thyroid hormone, what we call TSH suppressive therapy. As you know, many people who have thyroid cancer are given relatively high doses of thyroid hormone afterwards. And the goal of that is to make a hormone made by the pituitary called TSH as relatively low. And the reason for that is we don't want this hormone called thyroid stimulating hormone, TSH, to stimulate thyroid cells and to make them grow inappropriately. But it so happens that among these genes that I'm showing you here, one of them is called the TSH receptor. And it turns out that many patients have tumors that don't see TSH at all. So it doesn't matter whether or not TSH levels are high or low. The tumor cell has lost the ability to recognize TSH. And that tells us maybe those patients are not the ones that we ought to be giving high doses of thyroid hormone, which has side effects like arrhythmias and sleep disturbances and can lead to osteoporosis and so on. And we can begin thinking more in a personalized way, who should receive high dose of thyroid hormone and who can just be replaced at the normal level. So, what about therapy? First of all, what are targeted therapies? And the goal of targeted therapy is to kill or interfere with the growth of cancer cells without damaging the rest of the body, right? So we're trying to get a target sufficiently selective that it will help target the tumor and hopefully minimize the effects on healthy parts of the body. And there are many ways of doing this. One is to exploit a property of the cancer cell that is unique to them. And in the thyroid, we've been the poster child of this, in that radioactive iodine, in fact, was the first, or one of the very first targeted therapies. It's been in use more than 60 years. Okay? At that time, we didn't think of it that way, but of course, there are very few tissues that trap radioactive iodine, the thyroid being the dominant one. You can also try and target a cell surface protein, something that's on the outside of the cell, to which you can direct toxic antibodies, for instance, and there's a lot of interest in this approach for treating cancer. Or you can block the activity of these abnormal oncoproteins with small molecules, and that's a burgeoning and expanding field. And finally, and this is important because in the thyroid cancer world, this now has become a very dominant mode of treating the disease, you can interfere with the blood supply to the tumor. So, just a couple of words on this, because the two drugs approved for thyroid cancer happen to be drugs that block the blood supply of these cancers. So if you think in terms of a tumor from its inception, starting as a single cell, then dividing into two, from 2 to 4, and so on, at the very beginning, these cells do not require a blood supply for their nutrition. They can just get it from the media around the cells. The interstitium is sufficient to keep those cells well oxygenated and healthy and metabolizing well. But when it gets to a certain size, it needs blood supply. And there are certain cancer cell types or cell types that actually have the property of being able to recruit their own vasculature. They essentially call in capillary structures, they come, they surround, and they begin providing blood to this tumor that expands and grows in that way. As the tumor gets larger, the blood supply, which is quite disorganized, is very chaotic, but it actually sort of serves the purpose of the cancer to continue to grow. Now, the way in which these capillaries are recruited, in large part, is due to a specific family of receptors called vascular endothelial growth factor receptors, VEGF. And it turns out that the two drugs approved for thyroid cancer primarily work by inhibiting these receptors, these VEGF receptors. And they're called sorafenib and lenvatinib. Both are now in the marketplace. Both have had phase 3 registration trials. They work differently. There's a lot of excitement with lenvatinib that was just approved because the response rates were very high and because the duration of the response rate was also higher than what we had seen previously. They have slightly different patterns of side effects. They need to be individualized depending on the patient. Not everybody with advanced disease needs these, but those that do, actually both of these are viable options. Now, we have been interested in trying to improve the ability of radioactive iodine to treat patients who may be at an earlier stage of their disease, where perhaps the tumor is either confined to the local regional sites, or the tumors travel to a different part of the body, to the lungs or to the bones, but it's not growing rapidly. But still, we would want to shrink it down and prevent it from progressing. And what we have found is that radioactive iodine is often not very satisfying. It's not that effective in many patients with advanced disease. And if you remember that little slide I showed you with the green and the red, we now understand why. These BRAF tumors in particular just don't have the wherewithal to trap radioactive iodine. So I'm running a little short of time, so I'll try and go through this relatively quickly. We explored this in mouse models first. We made mice that had thyroid cancers driven by this BRAF oncogene. And when you do that, what we did, we were able to do it in a way that we switched the activity of BRAF on and off. And what you're looking at here is the iodine uptake of the thyroid of these mice. So if the thyroid is normal, is not expressing BRAF, then it traps iodine very well. If we activate BRAF, the expression goes down, the activity of the symporter is down, and then if you switch it off again, it comes back. And that we found that if you use drugs that block this BRAF signaling pathway, you can actually achieve a similar effect. And based on this, we designed a clinical trial. Dr. Larson was a very important part of that, and so together with many different collaborators at our institution, and this has become quite a well-known study. It was published in the New England Journal of Medicine a couple of years ago. And what we did is we used an inhibitor of many of these genes that I've already shown before are involved in thyroid cancer. So what you're looking at here is this little signaling pathway, which is essentially a message relay that happens from the cell surface, where there are receptors for growth factors, to these different proteins that in a way propagate the signal. And if you remember, there's mutations in these receptor tyrosine kinases, in RAS, in BRAF. So we said, well, if we have a drug that works distal to all of these, maybe we could actually get radioactive iodine incorporation to increase again. And that was the idea behind this. And we used this iodine-124 PET imaging that Dr. Larson's group had actually optimized at Memorial Sloan Kettering so that we could really detect the changes. And these are some examples. I don't know if Steve showed this before. This is one particular patient who we did an I-124 scan. And what you're seeing here is just the normal uptake of iodine in normal tissues. These are the sinuses and the salivary glands. This here is the gastrointestinal tract, the bladder, and so on. But unfortunately, this patient has metastatic disease from thyroid cancer, but none of these metastases are incorporating iodine. But after we give this drug selumetinib, now, yes, you start seeing these sites in the lung, in a lymph node, that are now trapping iodine. The same thing here we did in this patient that had metastasis to the lung. This is like a computer tomography image combined with the PET scan imaging or the iodine imaging. And you can see that before we gave selumetinib, some of these lung metastases did trap some iodine. But after we gave, it was much higher, and many additional sites were now trapping iodine. The same thing in this patient who had this little hole in the pelvis. That's thyroid cancer in the pelvis of this patient. And then when we gave the selumetinib, suddenly now it traps large amounts of iodine. And the same thing in the skull. This is at baseline. This is after selumetinib. And this is the same patient, but now just looking at the image differently. You can see this little area here. This is a skeletal metastasis in the skull of this patient. Traps a little bit of iodine, but not enough to treat. This is after the selumetinib. So this study actually benefited patients because it was a pilot study. It was like what we call a proof of principle. And we could show that many of these patients who did not respond to radioactive iodine before, when we give them this drug, their tumors really did shrink. So based on this, there's now a phase 3 registration trial that is at many institutions led by Dr. Alan Ho at Memorial Sloan Kettering, but it's an international study happening all over the world. And what we're doing here is in patients who have thyroid cancer surgery, and their thyroid cancers are actually pretty large, or they have large lymph nodes, we then randomize them to receive this drug, selumetinib, before radioactive iodine. Some patients receive placebo, because that's the way we learn if it works or not, and we do this on a 2-to-1 randomization. And that's an ongoing study that hopefully we'll learn if it worked or not in the next, I would say, year or two. So I'd like to stop here. I'd like to mention the people that participate in this, because just to give you a sense of what a large effort it is to put these type of programs together. I haven't discussed much of the activity of my lab, but I do want to acknowledge the people involved in this trial that I mentioned, and Alan Ho, many of my medical oncology colleagues. I mentioned Steve Larson, who's here, and his team in the nuclear medicine department. And then I think I'll stop here and happy to take questions.